Hello students, today we are going to discuss about the biosynthesis of RNA. Into the last lecture we have already discussed about the structure of RNA polymerase. Into the prokaryotic cells, the biosynthesis of biosynthesis of RNA is occurs in case of the biosynthesis of RNA occurs or location is cytosol. So what is mean biosynthesis of RNA? Biosynthesis of RNA is commonly known as a transcription. The specific double-stranded DNA get converts into the single-stranded RNA. It is a double-stranded DNA and it is a single-stranded RNA, and this process is known as a transcription. Into the process of transcription, the specific important enzyme responsible for the biosynthesis of RNA, that is RNA polymerase. RNA polymerase, it is a core enzyme that consists 2 subunits of the alpha. Also, the specific subunit consists beta prime and omega. It is a core enzyme that consists the 2 subunits of alpha, 1 beta prime and omega subunit. So the specific RNA polymerase, it is bind to the promoter region. The specific important region responsible for the binding of RNA polymerase, that is promoter region. This promoter region, it is a specific binding region for the RNA polymerase. So the specific, in case of the prokaryotic cell, the RNA polymerase, this is unable to bind to the promoter region. So what is mean promoter region? It is a specific region that consists of minus 35 to minus 10 region of double-stranded DNA. Minus 10 to minus 35 region of the double-stranded DNA is called as promoter region. At this region, consider where it is a minus 10 to minus 35 region. It is a plus 1. Plus 1 in this sense, it is a specific initiation site where the specific exact fragment of DNA, they will be responsible for the synthesis of specific RNA and proteins. Before that, the specific plus 1, it is a specific exact beginning region of the coding DNA. Before that, that is a minus 10. This is a minus 35. In this specific plus 1 site where the RNA transcription happens. But before that, minus 10 to minus 35 region, this region is known as a promoter region. That is a PR. That is a promoter region. And this promoter region is responsible for the binding of RNA polymerase. So the specific RNA polymerase, they have ability to bind to the promoter region. This promoter region is situated at the minus 10 to minus 35 region before the 10 subunits early of the initiation site. And from the plus 1 site, the specific RNA transcription will be occurs. Okay. So the specific RNA polymerase, they will be bind to the promoter region, and after the binding, they will be start the synthesis of RNA from the plus 1 site. But the specific RNA polymerase can't recognize the promoter sequence. The specific RNA polymerase can't recognize the promoter region. In this situation, the specific factor is responsible for the binding of RNA polymerase to promoter region, that is a sigma factor. So, RNA polymerase can't recognize the promoter region. So, to overcome this problem, the specific fragment responsible for the binding of RNA polymerase to the promoter region, that is a sigma factor. So what is the what is the function of sigma factor? The sigma factor is responsible for recognition of of RNA polymerase to promoter region. The specific sigma factor is responsible for the recognition of RNA polymerase to promoter region. So the RNA polymerase here, to the next question. Plus sigma factor is called as a holoenzyme. So the RNA polymerase, it is a core enzyme. This core enzyme plus sigma factor, it is considered as a holoenzyme, and this complete enzyme, they will be bind to the specific region of DNA fragment at the -10 to -35 region. That is -10 to -35 region. It is exactly binding site of RNA, and here a +20, here a +40. It is a -40, -20, and it is a holoenzyme that consists of sigma factor and RNA polymerase. They will be bind to the -10 to -35 region. That is a 5' to 3'. This is also 5' to 3'. And the specific region is called as Pribno box, and -10 to -35 region is called as a promoter region. So the specific -10 to -35 sequence is most important in binding of RNA polymerase. The -10 sequence is TAT AAT, and -35 sequence is 5' to TTG ACA 3'. and at the +1 position always adenine residue, always adenine residue is present. Adenine residue is present. So the -10 to -35 is referred as consensus sequence. Referred as a consensus sequence. So this specific sequence is important into the binding of RNA polymerase to the specific DNA fragment, and this DNA fragment they get converts into the RNA. So the specific RNA polymerase, they have this one N terminal, another one that is a C terminal domain, because that consists the amino group is as well as carboxylic group, and this C terminal or the specific CTD or C terminal domain, they have the specific they will be bind to the up element at the beginning portion before the sorry, it is not a +40, it is a -40, beginning to the -40, that is the up element, and to the specific, at the specific up element there it is a holding of CTD domain or CTD. So into the present lecture we will be discussed about biosynthesis of RNA. The location, it is a cytosol. So what is meant transcription? So transcription, it is a double-stranded DNA get converts into the single-stranded RNA by the process of transcription. The specific or important enzyme responsible for the transcription, that is the RNA polymerase. It is a core enzyme that consists the 2 subunits of the alpha, beta, beta prime and omega. So what is mean promoter region? The promoter region is the -35 to -10 region of double-stranded DNA is called as a promoter region, and it is important for the binding of RNA polymerase. So this promoter region, it is a beginning site of beginning or initiation site of RNA transcription or RNA biosynthesis. But RNA polymerase can't recognize the promoter region. So to overcome this problem, the specific factor is responsible for the binding of recognition of RNA polymerase to the promoter region, that is a sigma factor. They can play important role in the binding of RNA polymerase to the specific site, or that is a promoter region. So the RNA polymerase plus sigma factor is equals to holoenzyme. This complete or holoenzyme, they will be bind to the promoter region, or this promoter region is also called as a pribno. Or why should it is called as a pribno? Because it is firstly observed by the scientist Pribno, so it is called as a Pribno box. So what is the sequence at the -10 region? -10 to -35 sequence is the most important binding of RNA polymerase, that consists 5' TAT AAT and -35, that is a 5' TTG to ACA, and at the +1 position always a adenine residue is present. So -10 to -35 is referred as a consensus sequence, and this sequence is important in binding of RNA polymerase to the double-stranded DNA, and this double-stranded DNA get converts into the RNA by the process of RNA transcription or RNA biosynthesis. So details of initiation, elongation, the specific transcription is process of transcription is classified into initiation, elongation and termination. The specific process of transcription is classified into the initiation, elongation and termination. So initiation, elongation and termination will be discussed into the next lecture. So thank you.